a little bit with clinicians type, that what GWAS means to clinicians. But after my first talk a couple of hours ago, I thought that just to try to make a khichdi will be awful. So I will, because of the time constraint, I will jump some of the slides in this venture. What are the genetic disorders? Here I will skip some of my slides, because as a physician I am sure you understand about those genetic diseases, since the first genetic disease starts with a royal disease, which is a Mendelian monogenetic disease, that is factor VIII deficiency or hemophilia. So we are talking here about those multigenetic disorders, probably environmental related multigenetic disorder, not the monogenetic disorders. So this slide is depicting that what is the continuous trait and discrete and simple versus complex. Here if you have the mutation or single nucleotide polymorphism in certain gene, then you are going to show the pathogenicity or that your disease pathology precipitation. But here we do not know that whether multiple genes alone or there are other components are also there. More and more our modern research is pointing towards the figure that this type 2 diabetes and these lifestyle diseases, actually we are making this disease. Body actually when at the birth is not for the disease, because we are taking unhealthy lifestyle, and with some genetic signature probably we are proceeding towards this disease. So before going that, I am not going to this anymore, because if we see the Mendelian disease and multifactorial diseases, these are fully penetrant. You have the mutation, you have the disease, and these penetrants of the disease genes are going slowly vanishing, and in this end all of those asthma, type 2 diabetes, metabolic syndrome, those are the genes. This is the story so far already being described by my previous speakers, specifically Dr. Sridhar, that this is that actually that as a physician you have all of those organs. So those endocrine organs, your endocrinology, those rheumatology, they are taking some organs, those GI specialists, they are taking some organs. So this James Watson and Crick, they gave actually to the physicians one organ, that is that all DNA. So future of one of the chapters in your MBBS or MD eventually will be these genes, which started in 1953, and in 2000 the first human genome came, and after that these new technologies are coming where we can visualize or we can see, we can ransack genomics and epigenomics of all of those complex diseases. How do we know the trait is genetics? First thing is like those familial aggregation, and most of this data is coming from the twin study, monozygotic and dizygotic twins, and recently with the adoptive kids also we can tell something about that whether the gene is genetic or not. How do we search the genes? Before this new generation sequencing technology or GWAS, there were all of those gene hand for disease, specifically the family linkage studies. All of those earlier monogenetic disorders came through this way. These are some microsatellite markers and big families. And then you can fish out those genes, and all of those hemophilia genes and other monogenetic disorder genes comes with these family linkage studies. But with the modern, these complex diseases, this is to some extent is total failure. And these associations are like very simple way, cartoon picture, if you see this is the disease population and this is the control, and you are saving the single nucleotide polymorphism. If you see that disease are having this allele more, with the statistical help you can say this is associated with the disease. These are the candidate gene studies. And what is genomoid studies? Genomoid studies is actually those SNPs, but in many numbers, in millions. So instead of doing only one SNP at a time, now you can do 5 millions at a time. And then with the power of computer and the power of statistics, you can assess that these are the genes which are associated, which showed association with these sets, and you do a replication phase. And replication phase classically must be at least if it is n, it should be n plus 1. And then again you do individual statistics and then combine statistics to get some of those genes which are associated. It is just like that. So that candidate gene, you have a marker and you lost the key. So if it is not appropriately next to this light post, it's very difficult for you to get the key. Then you will have to do the fine mapping of that region. This GWAS is like those same light posts. They are placed individually in similar distance apart. So it's a light and street. If you lose a key here, again you will have to do a little fine mapping, but you are more nearer to that key. So this is the genetic testing of those candidate gene approach, where we got lots of monogenic disorder genes, and this is the modern approach where we are getting unknown multifactorial gene. And there is another difference. This candidate gene is hypothesis driven. When it was hemophilia, people thought that that must be clotting factors, and then they sequenced all the clotting factors available and saw that factor 8 is responsible and 9 is responsible. But here this is hypothesis free. That means if body has 5 million SNPs, you take all 5 million SNPs and then assessed, you can come out with the unknown genes or gene of new functions or certain new biology. Say, looking for diabetes, if you find that associated gene is a neurogene, then it will force you to think about the new biology of the diabetes. It's not the trios of liver, pancreas and skeletal muscle. Actually, the story of Indian type 2 diabetes GWAS is such, I will show you in a minute. So, if we see the power of the GWAS, how it is like that. If we start from the 2000, there is only gene discovery, that was the PPR gamma. And then before the 2003, we got KCNJ11. And then in TCF7L2 came in 2006. These are all candidate genes. Then with the invention of new tools, all of a sudden we find these all human genome related new toys. These are the new generation sequencing machine. This is GWAS reader, all of those new techniques. And all of a sudden in 2007, you will find there are lots of 21 variety at a time. So it gives you all of those new genes and new biology in a platter. And this is the current scenario of the chromosome. If you take the GWAS, all of those discovery over all of those diseases, complex diseases, it looks like that their all chromosomal positions are full with those some susceptible genes. And why we do GWAS? We do GWAS because of this. Because modern medicine gives two important things. One is antibiotics, which works miraculously well. We have some hurdles now. And second is surgery. Why antibiotic works? Because we know the origin. So it's easy to kill the origin. Here we do not know. So we are looking for pre-diagnostic marker just to give the warning to those would-be patients that follow a good lifestyle. It's unbiased approach, whole genome coverage, and may give a new biology. Let's see the first type 2 GWAS in Indian population. This based on those 610,000 markers on Illumina platform. We started with 2,500 case versus control of North Indian origin, and we got 10 to the power minus 3 significance, all of them, and then did one customized chip and tested them on almost 10,000 people, case control in 2 different cohort. This is North Indian cohort, is Dravidian cohort. And then these are those already being done and discussed, that how you select your case, how you select your control, and these are wonderfully characterized by all of those biochemical parameters, so that now we are doing quantitative trait locus of all Indian population. Now we know what are the genes control these phenomena or these biochemical phenomena in them. I will not bore you with all of those statistical QCs regarding that, but this is our GWAS. Every color depicts one chromosome. We got that TCF7L2 is already discovered, is the highest marker and highest significant marker in our population, but we got in a second chromosome a gene named TMEM163 as a very strongly associated in Indian diabetic type 2 population. What is that? After doing that we did one other technique called imputation. I'm not going into the detail, it's very simple from this cartoon. If you have this your reference panel and this is your data, so it's very simple. You can say all of those missing data will be A or T. So it gives you high, more number of markers. So in our case, is actually this is the reference panel 1000 genome, and this is our data. When you input these things there, we have now many new markers. So this is very important for those experimental purposes. And when we did that, we find this region where all of those highly significant genes are there. You know, all of these genes are associated with some of the neurological functions. They are established neurological genes. That's a new thing for us, and it forces us to think about the new biology. This is our GWAS, and that's we published that couple of years back. This is the largest genome-wide association studies of any genetic disorder from India. Now, what we got out of this GWAS? I'm talking about this, our gene is a synaptosomal membrane protein. So, when you know about the genes, in the worst dream of yours also, you cannot connect this with the type 2 diabetes. So this is the power of genome-wide association studies, is force you to look at the disease with a new biological angle, which eventually may give a new drug target. Now we know, after working with this TMM163, which is a zinc-binding synaptosomal vesicle transporter protein, we started working in the animal model, and we saw this protein expression is very high in human pancreas. But it was eventually earlier discovered at least 10 years back as a synaptosomal membrane protein. And after doing some of those other experiments, we also know this protein is helping in the endoplasmic reticulum to hold the insulin, and during the signal it helps to release the mature insulin into the bloodstream. That's another story, but this is the first time we are showing this type 2 diabetes has a brain angle. That means it's neurally related. So that's the power of those things. Now, what is these genetic signals mean? That these are those, somebody asked me a question that when it will come to you, to clinicians for use. Actually, we are not yet ready. But if you see this slide, forget about these things. These 10 genes are strongly associated with type 2 diabetes anywhere in the world. And these 10 SNPs means there are 20 alleles, because human chromosomes are biallelic. And you see this gentleman, 57-year-old, if you see this entire story, fasting blood glucose 221, HbA1c 10.5, you know this person is diabetic. So if we type these genes there, we find 14 of these risk alleles are here with this gentleman, only 6 non-risk alleles. Now you take this young kid, 15-year-olds, BMI wise a little obese, fasting blood sugar is 78. We cannot put that person as a diabetic yet. We measured these genetic things again, and we find this guy also has a 14 risk alleles and 6 non-risk alleles. So up to this point I can say that using these genetic markers along with these classical markers can make your prediction a little bit robust. Up to this point we can use this data until and unless we prove and we find out the causal variants of the disease. I think I'm done, and I can take any of those questions if you have any. And this is that our groups where we are doing all of those students are doing wonderful work. I have started with Professor Nikhil Tandon as the first, because when I was naive to diabetes and genetics, and after that lots of my physicians friends are there, because it's a large study, it needs lots of people involvement, and my money is coming from CSR mainly, and some money I'm getting from DBT.